Okay, what I want you all to think about is how much you've heard of antibiotic resistance. It's everywhere, right? But what is less well known is that a lot of these resistant infections are actually on medical devices. So what happens is a patient will go and have maybe a serious hip implant or a minor IV drip, and a bacterial biofilm will develop on the surface. Okay? A biofilm starts off life as normal bacteria, so that's how it gets into you. It's bacteria, they usually can swim. They have these kind of tails, and when they land on the surface of the false hip, for example, we think they use that tail to push fluid around themselves, and that sends messenger molecules out to other bacteria on the surface. And this communication stimulates them all to produce a kind of slime. The slime sticks them to the surface, but it also protects the bacteria. So that's all a biofilm is, it's just slime with bacteria in it. But the thing is that this protective slime means that things we'd usually use to kill bacteria, like antibiotics, don't work as well. So this is why biofilms are the leading cause of antibiotic resistance. They are everywhere as well. We're just learning now that 95% of all bacteria in the whole world form in a biofilm. So they can be the deadly dangerous type that form these infections. They can be the everyday type, the slime in your shower, or the plaque on your teeth. And they can be really useful too. We use them to produce biofuel and treat wastewater. So what I'm telling you is crucially important we understand how these biofilms form so we can control when and where they form. That's where I come in. I'm looking at how the bacteria communicate, because if they can't communicate, they can't form a biofilm. So I want to know what that flow the bacteria make around themselves looks like and how that helps them communicate. So I have an experiment with a bacterial cell on a surface pushing fluid around itself. And what I've done is put loads of tiny beads into that fluid. I'm going to follow what the beads do, and that's going to tell me the way the fluid is moving. Now, if I looked at this with a normal microscope, I'd only get 2D information. So I'm using a really exciting technique called holography, which is basically like a microscope but with lasers. So the laser light comes in, hits a bead and scatters. And that's what you can see behind me. You can see the rings that are formed. Now, these rings are different sizes depending on where the bead is in 3D space. So I can work out the whole 3D motion of the bead and therefore the 3D flow of the fluid. And what I found is there are certain areas of the surface that are more important for this shape. So we can start to think about developing a surface for a false hip, for example, that disrupts this flow and therefore disrupts the communication and a biofilm can't form. What I want you to take away is biofilms are made up of slime and bacteria. They are everywhere and they are so important to our lives. And that surface structure could be the key to their promotion or prevention. Thank you.